Um, um, Many thanks to the organizer of this symposium for giving me the opportunity to share our work with all of you. My name is José Antonio Sánchez Alcázar. I come from Pablo de Olavide University in Seville, Spain, and I'm going to try to explain a little bit the work we have done during the last 5 years on KAT6A and now on KAT6B. So I will try to explain the experimentation um, to make easier what we do in the lab. Of course, I and my group are basic research groups, although we always try to make a kind of research that can be translated to the patient as soon as possible. But we are basic researchers. We are not clinics. Um, so brain development is a tremendously complex process in which I'm going to take the pointer, laser point. So it's a very complex process in which many uh, cell types, neuronal and non-neuronal, are generated, assembled into functional circuits in a very high organized manner. Um, neurodevelopmental disorder can be caused by many uh, causes, postnatal causes, perinatal causes, prenatal causes, and congenital. Um, we are in this meeting, we are more interested in the genetic causes, which can affect chromosomal modification, monogenic pathologies, polygenic pathologies, and also epigenetics, which are modifications and dysregulation of the way can control cell gene expression. So this epigenetics modification can be due to failures of genomic imprinting, X chromosome inactivation, a mutation of proteins associated with epigenetic gene regulation, such as KAT6A and KAT6B. So epigenetics is a complex process in which different mechanisms intervene, interrelated with each other to modulate gene expression without producing changes in the nucleotide sequence. The main mechanisms involved in epigenetic regulation are DNA methylation and also histone modification. And this histone modification can be phosphorylation, methylation, or acetylation. So to make something, this issue a little bit more understandable, you have modification in the DNA and also in the proteins which are near the DNA and can regulate the expression of this molecule. So, respect to histone acetylation, there are two groups of protein regulating this process. The KAT6A and KAT6B, which is acetylating the histones, and the sirtuins, which are desatylating the histones. So in this way, DNA expression can be regulated. And normally, when DNA is acetylated by KAT6A and or KAT6B, there is a increase in the transcription and in the expression of the genes. Both protein KAT6A and KAT6B are working in a complex which include several proteins. You have here the name of some of them, and all this complex is the responsible for the acetylation of the histone in the DNA. So when these proteins involved in histone acetylation are affected by mutations, they cause disease. In the case of KAT6B, it's called KAT6B syndrome, which includes two group of syndromes, the genitopatellar syndrome or the Say-Barber-Biesecker-Young-Simpson syndrome. Um, this mutation produce heart abnormalities, developmental delays, into intellectual disability, decreased muscular tone, genitalia anomalies, microcephaly, etc. When the mutation is in the KAT6A syndrome, they also when the mutation is in the KAT6A gene, they also produce a syndrome with very, in some ways, similar symptoms, developmental delays, intellectual disability, heart abnormalities, movement disorder, etc. They are both of them are autosomal dominant mutations, and there is not a specific treatment so far. And one of the most, noticeable properties of this syndrome is that there is a heterogeneous symptomatology. Here is the KAT6E gene, which is, affecting the process of the acetylation of the histone. Is regulating chromatin remodeling, regulation of gene expression, protein synthesis, and cell division. So the main problem is that the mutation in these genes, which are involved in the acetylation of these proteins, which are bound to the DNA, and when these proteins are not working because they have mutation, there is a lack of acetylation of the histone, and that means that there is a dysregulation of the expression of particular genes which can be involved in many important cellular processes. It has been postulated by Dr. Richard Kelly and others that this dysregulation of the expression of specific genes can affect an important organelle, which is the mitochondria, which are the powerhouse of the cell. So they produce the main part of the energy of the cell. So the defect of the expression of specific genes here in the DNA can affect secondarily the mitochondrial bioenergetics and the production of energy. So in our lab, we are a mitochondrial lab, so we were, very interested in going deeper in this hypothesis. So mitochondrial dysfunction due to the defect in the expression of these genes can induce oxidative stress because many of the electrons don't go in the right way and go to the oxygen and produce free radicals. So there is a increase in the oxidative stress in the cell, which can damage cell components and can induce including cell death. So cells also have a protective mechanism against these free radicals produced by mitochondria. They have many enzymes and antioxidant systems to try to defend the cell from these free radicals. So it's also important to know in KAT6A, KAT6B syndrome that the mitochondria is not working well, is producing free radical, and also how is the antioxidant system inside the cell, such as superoxide dismutase, catalase, or glutathione peroxidase. So the main question is that the deficiency of mitochondrial function can produce a defect in the production of energy, and for this reason can be a big problem for the developmental process and also for the normal function of many cells. Another aspect we are interested because we have studied some neurodegenerative diseases in which coenzyme A is affected, which is another very important cofactor produced from pantothenate in this very complicated pathway. But this coenzyme A can be produced in the cytosol of the cell, but also in the mitochondria. And the mitochondrial pool of coenzyme A is very important for not only the um, uh, putting the coenzyme A cofactor in this molecule, the acetyl molecule, but also for other processes which are essential for mitochondrial function. So we were also very interested in knowing how the deficiency in mitochondrial function is also affecting the coenzyme A metabolism in KAT6A and KAT6B. So just to summarize a little bit the key role of coenzyme A, I would say that it's important for amino acid and lipid metabolism. The the functioning of the Krebs cycle, protein modification, cytogenesis. So, and also is very important for the production of this molecule, which is a very important protein in mitochondria, which is mitochondrial ACP, which is involved in all this function in mitochondria. So in many of the experiments I'm going to show you, we have evaluated the expression of this protein because it's key for the production of energy in the mitochondria. So in summary, what we have to know is that these two proteins, KAT6A and KAT6B, are very important for the acetylation of histone in DNA. The defect of these proteins can lead to problems in many cellular functions as transcription, cell cycle, signal transduction, etc. And of course, it's affecting many developmental processes such as hematopoiesis, neurogenesis, skeletogenesis, heart development, craniofacial development, body segment identity, etc. So, how to study these diseases? So there are different approaches from our point of view. All of them are very useful because in science we think that any aspect is worthy to investigate. So there is models in mice, zebrafish, and in both diseases, in KAT6A and KAT6B. And they have, recapitulated many of the problems the patients have. Uh, in our lab, we follow another approach that we try to modelize the disease with cell cultures derived from the patients. So we use a skin biopsy. From this skin biopsy, we generate fibroblast cell cultures that is from our point of view, is a wonderful model because, of course, they have the mutation of the patients and also allows to do what we call personalized medicine because we have the mutation of a particular patient in a bottle. We can do and test everything in this particular mutation. I think that for all the genetic diseases, it's very important to have this model because you can, as you will see later, test the alteration and you can also evaluate which compounds can be useful for correcting the mutant phenotype. So from our point of view, it's key to have the fibroblasts from patients with genetic diseases such as KAT6A and KAT6B. The other advantage of this model is just because you can think that maybe fibroblasts, the cells of the skin, are a very humble cell compared to neurons. But the good thing is that now we can transdifferentiate the fibroblasts from the skin to neurons. That is supposed to be one of the main type of cells affected in this type of diseases. So we can have the mutation of a particular patient in the bottle, and we can transdifferentiate or reprogram these cells to neurons and see how is the mutation is working in this particular cell type. So in here you have the fibroblast cell cultures, and after 27 days of reprogramming, you have neurons. Of course, you check that these are real neurons because they have the expression of particular markers and also have electrophysiological activity. So with this model, we we propose to do personalized medicine for this type of diseases and many others because we work with neurodegenerative diseases, we work with neuromuscular diseases and mitochondrial diseases. In all of them, we follow the same protocol. We have the fibroblasts, and later we go to the reprogramming and confirm the results of the fibroblasts in the neurons. So this project of personalized medicine relies in the assumption that the different mutation can also contribute to both the susceptibility to the disease and to response to the drugs. So maybe some particular mutation can respond to particular drugs and other mutation can respond to others. The aim of this approach is to maximize the likelihood of therapeutic efficacy and to minimize the risks of drug toxicity. So with this model, we can do something that from our point of view is very important, is to know the behavior of a particular mutation and know which drug and which concentration is critical, which concentration can correct the mutant phenotype. So from our point of view, the information provided by genome sequencing, pharmacogenetic, proteomics, metabolomics, bioinformatics is very important, but also is very important the response of cells to different treatments. So it's a very uh, rational complement to decide what is the best option for therapy in this patient. So we started the project in 2019 with the KAT6A cells, and these are the objectives that basically were the characterization of the which alteration are found in the fibroblasts derived from the patient, to do a pharmacological screening based mainly in the supplementation with commercial compounds. The the use of commercial compounds is very important because can allow the translation to the patient more quickly. We confirm this molecular mechanism of these positive compounds, doing more assays. We assess the status of the acetylation in these cell models. We confirm also the potential treatment in these cells. And now I'm going to show you some results. Um, I I will explain a little bit about which techniques we use. This is a technique. This is called Western blotting. So we have here lanes in which you have these black lanes, and they show the expression of the protein. So we do this type of assays both in control cells and in patient cells, which are named P1, P2, and P3. And the controls, which are cells derived from healthy donors, are called C1 and C2. So in here you see the levels of the controls, and here the levels of the patient-derived cells. So in this way, we have an evaluation of what is the levels of the proteins in normal situation and in the patients. So in this experiment, of course, as we expected, the KAT6A protein is downregulated and is reduced in the patient cells compared to control. But also there is the regulation of other proteins which are important in the process of the acetylation, such as SIRT1 and SIRT3. So that means that in KAT6A mutation, both the acetylation and the acetylation process are modified and are dysfunctional. And of course, we check this protein I mentioned before, the mitochondrial ACP, because it's very important for mitochondrial function, and you can see how it's reduced in the patient samples. And also downstream proteins, which are dependent on the functioning of mitochondrial ACP, as lipoylated pyruvate dehydrogenase and also lipoylated another dehydrogenase. Both are also affected in the patient cells. So, and these also represent the protein expression of uh, proteins involved in mitochondrial functioning, different subunits of mitochondria, and all of them are downregulated in patient samples. So, as mitochondria is a very important organelle, which is also very essential for the iron metabolism. We also tested the expression level of many proteins involved in iron metabolism, and we found that they were altered in the patient samples, as you can see here. and also some proteins involved in the formation of the iron sulfur cluster proteins are also down regulated. Here are the proteins which are involved in the antioxidant system, which are also reduced in the patient samples. So, as you can see, there are many experiments just to demonstrate that there is an alteration in mitochondrial function and the antioxidant system. So later, what we did was to uh, design a screening platform for compounds for potential interest for therapy. So we put the cells in an stress medium with galactose and oligomycin, and in this situation, control cells survive, but the patient cells die. So as you can see here, almost all the cells die. So what we do now is just to add different compounds and see which one is rescuing the phenotype. So as you can see, cells die, and after pantotenate, the cells survive. Cells die, and after carnitine, the cells survive. So theoretically, with pantotenate and L-carnitine, we have a kind of uh, potential treatment for KAT6A fibroblast in this particular mutation. So you can quantify the viability of the cell cultures and see clearly that cells die, and after pantotenate or carnitine treatment, the cells survive. So we can also do a combination of both compounds and see that there is a kind of synergic effect. So you can see here how the synergy is has effect with pantotenates and carnitine in the sample cells. So with this way, we could do what we call personalized approach. So patient one needed 0.7 micromolar of pantotenate and 0.7, but patient two needed 0.4 and, of pantotenate and 0.4 of carnitine, and patient 3 is 0.7 and 0.7. So the question is that not all the patients respond in the same way, and that depends on the type of mutation, of course. These data are very important because if we know that the mutation is corrected with this concentration, from the clinical point of view, it's important to know if the dosification of the different drugs can reach this concentration. So cells give a lot of objective information about what is the right concentration must be reached in the patients. So later, as we knew that carnitine and pantotenate are able to rescue cell death in the patient cells, we confirm the result just checking all the proteins that were altered before. And the combination of pantotenate and carnitine, fortunately, were able to rescue the mutant phenotype in all these proteins that were affected in the patients, including mitochondrial proteins, protein related with coenzyme A metabolism, antioxidant system, all of them. So, and another thing we used to do is just to confirm the result with different ways. So, as is supposed to be an increase in acetylation of the acetylation after carnitine and pantothenate treatment, what we do is to study the acetylation of histone before the treatment and after the treatment. So we found this technique very useful to see clearly that a particular treatment, in this case pantothenate and carnitine, is able to increase the acetylation of the histone. And also we do it in all the patients, and it's not very complicated technique. So we also confirm these results with a colorimetric assay in which we measure the acetylation of the histone. Both carnitine and pantothenate are able to increase the acetylation of the histone. And of course, if mitochondria is are dysfunctional, we confirm that the treatment with pantothenate and L-carnitine was able to increase the bioenergetics and mitochondrial function by this assay, which is called a seahorse assay, in which we see basal respiration, at ATP production, maximal respiration, and spare respiration. All of these parameters give an idea of how mitochondria is working. And we did it in all the patients treated with, in all patient cells treated with pantothenate and carnitine. And also, you can see the functioning of the mitochondria just looking at the production of free radicals. And you can see here the patient cells have a lot of free radicals, and after the treatment, the radicals are eliminated. Finally, what was another important point is just to see if we can modify the transcriptomic pattern of these cells after the pantothenate and carnitine treatment. So we perform a RNA-seq in which we can see the transcription of all the genes. And with this type of graph, you can see genes which are expressed less and genes which are expressed more. So in different conditions, control, patient, treated patients with pantothenate and carnitine. So we can see how the treatment can alter the expression pattern in the patient and can increase and modify this pattern, being closer to control cells. So we confirm that many of the genes we detected in the RNA-seq were indeed upregulated after the treatment by PCR. We do this type of analysis in more patients, in 5 more patients from different countries, including USA. In all of them, we confirm that pantothenate and L-carnitine are able to correct the alterations that were detected in the cell. And just you can see here clearly how the histone acetylation is very low in the cell of this patient, and after the treatment, the acetylation is improved. So, in summary, KAT6A mutant fibroblasts show deficiency of the KAT6A protein, deficiency in protein related to acetylation and the acetylation, deficiency in protein related to mitochondrial metabolism, deficiency in protein related to coenzyme A metabolism, deficiency in protein related to the antioxidant system, deficient mitochondrial bioenergetics, increased ROS production, and altered transcriptomics. the pantothenate and carnitine, maybe just by a mechanism of increasing acetyl-CoA, which is the substrate of the enzyme, is able to partially recover many of the alteration we have observed. And also, we the, it's very important that the treatment is able to modify the transcriptomic pattern, especially because it's able to increase gene related to mitochondrial and coenzyme A metabolism, and also related to the antioxidant system. In conclusion, in this work, fibroblasts derived from patients with mutation in the KAT6A gene show a deficiency in protein related to acetylation deacetylation, coenzyme A metabolism, mitochondrial metabolism, iron metabolism, and the antioxidant system that are essential for proper cellular function. And the treatment with pantothenate and L-carnitine correct pathophysiological alteration at different concentration depending on the patient mutation. Treatment with sodium pantothenate and L-carnitine recovers the expression level of both the mutant protein and other protein related to mitochondrial metabolism. Histone acetylation and transcriptomic patterns are altered in fibroblasts derived from patients with KAT6A syndrome. The treatment with pantothenate and carnitine partially recover both histone acetylation and the transcriptomic pattern in fibroblast cell lines derived from the patient. And the different response to treatment with sodium pantothenate and carnitine suggests that personalized medicine approaches may be of interest in KAT6 syndrome. So we published these results, and later we started the project with the KAT6B fibroblast. The things are being a little bit more complicated because the fibroblasts derived from this patient grew more slowly. So we had some difficulties to do all the experimentation we wanted to do. But well, the thing is that we got 4 cell lines derived from 4 patients from Spain, and we started the experimentation. These are the mutation of KAT6B that we are studying right now. We have 3 that we know for sure that are KAT6B mutation, and we have some doubts with this one because the genetic report um, said that was a potential uh, um, pathological variant, but later when we started doing the experiments, we observed that there is no defect in acetylation. So in the histone acetylation. So we think that this patient is not a real KCSB. So, but we have these 3 cell lines from different patients in which we can observe the defect of histone acetylation, and also that pantothenate, especially pantothenate, is able to increase the acetylation in these cell lines, and also the combination of pantothenate and carnitine. So in here you can see how the treatment is also able to modify the behavior in this mutation affecting KAT6B mutation. Another important aspect, as I mentioned before, is the production of free radicals, which is affecting many molecules, especially we were very interested in knowing if we can detect lipid peroxidation in these cells. So as you can see here, are the lipid peroxidation in control cells, and these are the lipid peroxidation in patient F and also in patient O. So that this data show that patients manifest a lipid peroxidation that can be very important because lipid peroxidation is related with iron accumulation and can affect many other cellular function. So we would like to go deeper in this way just to understand better all the processes which are altered due to lipid peroxidation in KAT6B. Um, and here you can see how also many of the proteins that were reduced in KAT6A are also reduced in KAT6B, especially the mutant protein, which is here. This is the levels in the patient that is very reduced, but also KAT6A is reduced. I think this data is also important that when you have other downregulation of KAT6B, KAT6A is also downregulated, especially because they form this complex that uh, I explained before. And when one protein is downregulated, the other one is also downregulated. And also it's affected the other group of proteins, which are the acetylase function as SIRT1, SIRT3. So there is, in summary, this data show that in KAT6B there is a downregulation of both the acetylation and also the deacetylation of the the cells. Um, the good thing is that both pantothenate and carnitine is able to recover many of the alteration in protein levels we see in the patients, and also this important protein I told you before, mitochondrial ACP, which is very low in the patient, is recovered after pantothenate and L-carnitine treatment, and also proteins involved in the iron sulfur cluster biosynthesis, ferritin, and of course the mutant protein is also improved. The expression levels of this protein are also improved by the treatment, and we can quantify this. So we have many things to do with Praxis B. The experimentation has been delayed because the proliferative capacity of this cell is lower. So we have to continue the work. Um, so we are just looking for this new phase in which we can see the effect of this to study more deeply the how it is the mechanism of these positive compounds in the transcriptome pattern. We we want to generate neuronal cells by direct reprogramming of fibroblasts from these patients and to confirm the results in these neuronal cells. Also, we have to corroborate that positive compounds in fibroblasts are also positive in neuronal cells. So I think the platform for screening potential drugs is very useful for this uh, approach. So the project has been supported by public funding, but also for patient association, especially Superauténticos, with Águeda Pérez, KAT6A and Amigos, the Spanish association with Alberto Franzón, of course, KAT6A Foundation that always supported during the last years. So this is the group. We work with different mitochondrial diseases, neurodegenerative diseases, muscular diseases. They are a group very enthusiastic and committed with the research and rare disease investigation. So if you want more information about what we do, we have a web page. You can check our, publications and um, everything we do. We try to disseminate all the work for, uh, to make more visible this type of diseases and the research we are doing. Thank you very much.
Okay, does anybody have question? We do have time for one or two questions really quick. Sorry, let me turn the microphone. There may be feedback. We'll see. So you have mentioned that these KAT6B fibroblasts grow slower in cancer than the KAT6A. Do you have any hypothesis on why this is happening or could be expected?
Uh, well, I think that both. I think that they have more problems, maybe pathologically. Um, there are sometimes there are technical issues in the lab, um, and they sometimes the cells don't grow so fast. Um, I think it's a mixture of both. But actually, what I have noticed is that at the cellular level, KAT6B has a stronger pathophysiology.
Thank you very much. Did you observe issues with lipid peroxidation in both KAT6A and KAT6B, or is it just KAT6B?
Sorry?
Did you observe issues with the lipid peroxidation
Ah,
both
yes, it's the same in KAT6A.
in KAT6A and KAT6B?
Yeah, it's the same. Actually, lipid peroxidation is very important, especially because what we have found studying another neurological disease, neurodegenerative disease called PILAN, which is affecting phospholipase A2G6 gene. So uh, we made the conclusion that lipid peroxidation is connected with iron accumulation, and iron accumulation is connected with lipid peroxidation. So there is a what we call a vicious cycle. So when cells have lipid peroxidation, normally they accumulate iron. And when iron is accumulated, you have lipid peroxidation. So it's a vicious cycle. We published a review about this special thing, the vicious cycle of lipid peroxidation and iron accumulation. And I think it's very important for many neurodegenerative diseases because in all of them, there is iron accumulation or lipid peroxidation. So, and you never understood well why it's so connected, no? The thing is that lipid peroxidation normally affect mitochondria. So, mitochondrial lipids are very important for the function of mitochondria. And mitochondria is a very rich iron rich organelle. So if there is a dysfunction in mitochondria, normally you have a more concentration of iron in mitochondria that is not handled in the correct way, and there is iron accumulation in the form of liposuction. So we think that there is a connection between lipid peroxidation, mitochondrial dysfunction, and accumulation of liposuction. Actually, liposuction is a autoamputation of a dysfunctional mitochondria. So mitochondria release the oxidized material.
What do you see signs of that in your blood test, like acting iron in the blood? Are there ways that we should be screening our kids for that?
Uh, well, I know that there are a specific iron accumulation neurodegenerative diseases in which iron is accumulated in one specific part of the brain, for example, in globus pallidus or in cerebellum, in some others. In other cases, I think one uh, I think should be studied by MRAI if there is a potential accumulation of iron in this patient. Sorry, can you repeat? Well, from my point of view, I think that this there is a great variety of patients because some patients manifest autism, another manifest severe developmental delays. So the spectrum is very different in all the patient. So I think that this type of treatment can help to improve some of the dysfunctional neurons that, of course, are there. Of course, the treatment cannot do anything for the neurons which are dead or are disorganized in the brain of the children. But I think can do a good job in the way, for example, in I don't know, maybe Dr. Kelly has more experience of what is doing well the treatment in the patient. What I know from the Spanish experience is that the children are doing much better, especially in the concentration, in doing some abilities that they didn't do before. Um, I think the impression I got from, only the family comments is that the children are doing much better. Of course, if the child has a very severe developmental defect, we should be more prudent with what we expect.
My question is following up on the first question that you had, and I wondered if there were any other factors that may have influence the difference between the response you saw with KAT6B and KAT6A? Other factors such as were those patients taking any medications? Was there a difference in their age? Those sorts of factors.
I think it's very early to draw any conclusion for this work because we need to do more experimentation and to compare more KAT6A with KAT6B. Now we have the sample, so it's just a matter of time and people working in this particular issue. I think it's a very interesting phenomenon. Um, we can, of course, you can do many things. Sometimes you in the lab, uh, when you study something, there are many ways to continue for continuing the work. Sometimes we are under pressure, especially by the patient association to find something very quickly. And so, but this aspect of comparing the behavior of CAC6A and CAC6B can be very interesting. All for our point of view, many things are worth it for going deeper. Um, but sometimes we have to decide what is the best for to make up a publication as quick we can. Um, but it's true. This is interesting.
Thank you so much for the interesting talk. So my question is, you have presented a reduced amount of KAT6A or KAT6B protein is leading to the pathological phenotype that you observe in cell culture. I was wondering whether you have tried overexpression of the wild type copy of KAT6A or B in your system to see if you see any recovery of this kind of pathological observation that you've seen in this situation.
No, we didn't do this type of experimentation. I think the functional complementation in dominant mutation is more complicated to draw a conclusion, but I think it would be worthy also to do it, to do the overexpression of one of them. Um, but we didn't do it.
Yeah, thank you. So I would ask also, we would like to, welcome patient and family questions after the talks here, but for more scientific or research questions, we are going to have a 2 hour kind of researcher or meeting in the end. So if we could, for the sake of time, restrict the more scientific questions to that session, just to stay on agenda, I appreciate it. Thank you. Two quick announcements. So the blood draws that are available here for patient families, those are now available and can be done